Good afternoon, everyone. It's good to see you. Welcome to our environmental health sciences seminar series that we do. We try to do every month or so, and we are so blessed to have this opportunity. And I'm going to turn it over in a minute to Dr. Unrein, but I just want to, if you're just joining us, if you are a member, you know this, but if you're not a member, become one. But we have a research portal. If you need help services with your research, even conceptualizing research or analyzing samples, or you need help with design, that kind of thing, if you'll put a request in our online portal, and Carol will put it in the chat, that would be great. So I want to turn it over to Dr. Jason Unrein, many of whom you know. Jason is a wonderful colleague in the College of Agriculture, and Food and the Environment, in CAFE. But he also leads up our UK Cares Access core, which is our analytical core. So Jason, take it away.
So it's my pleasure, everyone, today to introduce Dr. Sarah Jovan. Sarah is a research ecologist and the lichen program lead at the U.S. Forest Service Pacific Northwest Research Station in Portland. And she's been there since 2010 in that role. Her research is on the use of lichens and bryophytes, which include mosses, for bioindication of air quality and climate change. So some of the projects that she's working on are looking at nitrogen, sulfur, and metal deposition near oil and gas drilling sites, lichen-based critical loads for nitrogen and regulation of air quality in our national forests, using lichens as indicators of climate change in Alaska. And then what I thought was really sort of germane to our work at UK Cares was her community engaged work using lichens as air quality bioindicators in urban environments. So Sarah did her BA in biology at Oberlin College in Ohio and her PhD in ecology at Oregon State University. So just to give you a little bit of background, so Sarah and I have recently established a collaboration, and we actually have a new postdoc who's an analytical chemist, Dr. Jalili Jahani, who's on the call, to sort of help her with some interesting analytical questions related to her work. But I first found out about her work by reading an article in the New York Times about her work in Portland, which I think maybe she'll talk a little bit about today. And one of her collaborators on that work was Patrick Ryan, who's at one of our sister environmental health sciences core facilities at the University of Cincinnati. And so I was very interested in the work that they were doing and emailed them and ended up collaborating with Patrick, and we published a paper out of that. And then now have established this collaboration with Sarah. So I'm so glad that I sent that email, and it's led to a lot of interesting work. So with that, I'm just going to go ahead and turn it over to Sarah to have her tell us about her research.
Great, thanks for having me. Let's make sure I know how to advance this. Front. Okay, I think I got it. Yeah, so thanks for having me. We're going to talk about moss and lichen bioindicators of air quality. So just a quick overview of what we'll talk about. So we will start with a brief discussion of using moss and lichens as bioindicators. And then we're going to dive into the details of three very different case studies where we use moss to visualize fine-scale deposition patterns and heavy metals in urban areas. So we'll put a lot of time into describing study impacts and how engage the community and other partners in the area we're in the research, as well as some lessons that we learned along the way. There you go. So I just wanted to say up front that I'm not by any means an expert in like human community engagement in scientific research. I was trained in botanical and ecological sciences, specializing in lichen community ecology, which of course is very different. So I've sort of accidentally fallen into community engaged research, and I'll explain how that happened. But I speak mainly from an experiential and not a technical perspective on that element of the work. But it has become a pretty critical part of how we're doing these studies. So just a quick review. What is a lichen? So a lichen is a symbiotic relationship between a fungus and one or more photosynthetic partners. There's actually a Nature paper a few years back that suggested there might be even more kinds of symbionts that live within lichens, like yeast and such, if I recall. Anyway, so the joke about lichens is that they are fungi that have discovered agriculture, and they come in all these beautiful, very different looking forms. So moss, on the other hand, is really, a primitive plant, the oldest plant lineage actually, and the first to make the transition from, the primordial soup to growing on land. So this photo actually is from the forest floor in Boreal, Alaska, where you can practically hear the feather moss and the peltigera or dog lichen duking it out for space in this waterlogged muskeg. Anyways, my point is, while these organisms come from completely different kingdoms and are just so different in many ways biologically, they interact with the environment similarly. So they're often seen competing for space. But this also means, we can use them for air quality bioindication in similar ways. So both organisms are non-vascular. So that means that they lack any specialized structures that help them transport water. They also lack roots, and they absorb all their nutrients in their water from the atmosphere. So coincidentally, pollutants can also be absorbed and accumulate in lichen and moss tissues. So moss leaves are actually one cell thick with nutrient and cation binding sites on their cells, just exposed to the atmosphere. This is one of their adaptations that allows them to efficiently grab whatever nutrients come their way. So passive absorption through those sites is one of a few ways that moss take up pollutants. There's other mechanisms too. For instance, they have a high surface area and all these little side branches and leaves and such. They're also very good at trapping particulates and such, for instance. So there's two ways that we can use moss and lichens as bioindicators. The community method is very helpful for investigating pollutants that can cause significant ecological harm or change, like excess sulfur and nitrogen. So we often use this approach in studies focusing on environmental health, where the anticipated end user of the research, besides other scientists working on bioindicator methods, are federal land managers. So excess nitrogen, for example, can cause these dramatic shifts in community composition where sensitive species are lost. Those sensitive species tend to be the native or endemic species that play important functional roles in the ecosystem. You know, at the opposite end of the nitrogen gradient is an increase in these species that possess more weed-like traits. You know, they tend to be small and very fast-growing, and very widely distributed geographically, so they can rapidly colonize and benefit from this excess nitrogen. So it's similar to the sort of moss or lichen flora that you'd naturally see, for instance, around like bird perches or other areas where there's wildlife waste or something like that. Oops. So it's been about 10 years now, though it still feels like a recent change. You know, we've started focusing on urban forests where, you know, communities are often too impacted to really be very informative about air quality. So in these circumstances, you know, if you have at least one persisting lichen or moss species with reasonable abundance, you know, you can measure concentrations of pollutants in their tissue. Or if you don't have any moss or lichen growing in situ, you can use transplants from cleaner areas, but that is a whole other Megillah. We are going to focus, at least for this talk, on using in situ material. So I just have to say the Europeans have been doing studies like this since the 70s. So, you know, we didn't discover it, but, you know, we have made some innovations in study design, which we'll talk about in just a bit. So far, we've mainly focused on extracting heavy metals from moss and lichens and some associated elements, but and we can do that in-house. We do have a lab in Grand Rapids, Minnesota, where we have a lab manager, John Larson. We call him the moss boss because he runs all of our moss samples for most studies. Anyways, there are a lot of other studies that do show you can use moss and lichen, you know, to extract other kinds of pollutants. So a very interesting tool. So let's just move on to our case studies. So our first example was in Portland, Oregon in 2013. So about 10 years ago now. Doesn't feel like that long ago. And so this was our first foray out of monitoring forest health and into, you know, like human habitat, which is just an entirely different ball game. So we adapted our forest monitoring protocols using moss and lichen tissue to fit the urban environment. And we didn't have a sense actually that air quality was a particularly big issue in Portland besides, you know, PM from smoke. It was sort of just the set of circumstances that led us to a pilot project here. You know, research and travel funds were really low at the time. And so doing this study in our own backyard was very cost effective. We knew that there was only one permanent air monitor in Portland, so that's this star here on the map, which is actually a pretty typical situation when it comes to networks that monitor air toxics. We also noticed that there was this stress tolerant moss you see in the picture here, Orthotrichum lyellii, that just grows abundantly pretty much everywhere on street trees. So we were able to measure pollutants in 346 moss samples collected across the city for arguably about the same price as the monitor measurements taken for 1 year. So you can see our sample sites on the map. We had divided the city up into a 1 by 1 kilometer grid, and within each grid cell, we randomly selected a residential address, and we went there and collected the moss. And we also did some replicates, etc. So moving on. We did end up measuring a bunch of pollutants in the moss. And we're going to focus on the heavy metals. Granted, initially we were more interested in the PAHs, just because less people had done it. So we thought that that would be the more interesting part, which it wasn't. So, you know, our goal at its very simplest was the creation of data products using bioindicators that would really help decision makers, that would ultimately lead to consequential change. And I don't mean that in an activist-y way at all, but, you know, consequential change in a regulatory or perhaps at a public health level. You know, there are a lot of lichen and moss studies before us, including some of my own, and, you know, they have these beautifully crafted recommendations that languish in scientific journals, that, you know, decision makers never see. And so that's been quite a habit in this field for quite some time. So we needed to do a better job connecting the dots. So, you know, partnering with the stakeholders, you know, most likely to be able to respond to our findings. I know this is not an Einstein level epiphany, but as I said, that just wasn't really happening in this research field. So, you know, for this project, we got in touch with the Oregon DEQ, the Department of Environmental Quality, right away. And they were very interested in what we were up to. And they ended up being a pretty critical part of the study, as you'll see. So what we're looking at here is the DEQ was first and foremost very interested in our cadmium data. So this map shows their anticipated atmospheric cadmium concentrations that they estimated based on dispersion modeling, which is based on simulating how pollutants disperse from known emission sources and then estimating their concentrations at different locations using that model. And this is a very common approach used to make, you know, nice looking, but ultimately sometimes not very accurate continuous maps of air toxics. You know, as I said, air quality monitoring, direct monitoring is very expensive. So I'm not, you know, trying to attack the DEQ or air regulators for using this tool. It seems like an interesting part of an investigation. Anyway, so what drives me nuts about it is that it's very deterministic. For instance, you know, what happens if you aren't already aware of an emission source? So I'll answer that question in a second. So to the DEQ's credit, you know, they noticed that cadmium, they predicted using their emissions-based model was an order of magnitude less than cadmium measured at Portland's one monitoring site. So they knew that something was up, but they didn't know what to do about it. So we actually had pretty good timing. Okay, so if you look at their map on the right compared to our map of cadmium and moss on the left, you can see they're pretty different. You know, in the moss map, you see we have these two cadmium hotspots. A larger one here in the south, which we'll examine much more closely in a moment. You know, our geospatial model showed that these hotspots were very closely correlated with two stained glass manufacturers who are using cadmium to produce red glass. The pattern basically had zero correlation to known sources, which are indicated by these purple circles. So, as you know, we measured a lot of elements in moss, and so we looked into stained glass manufacturing and learned that arsenic and selenium are also sometimes used. So these are just some basic Kriegs based on our arsenic and selenium concentrations in our moss samples. And so you can see around the second larger facility, you know, an arsenic hotspot as well as a selenium hotspot. So, you know, on paper, this was an elegant example of using moss as a screening tool. You know, moss is inexpensive. We can sample quite densely. You know, it can't tell us directly about human health risks, but it can help us place those rare air monitors in places that could be problematic, you know, and therefore allowing us to use those limited resources more effectively. So DEQ installed a monitor across the street from the larger facility. And so here's just a map of the area. So here's the glass facility, here's the monitor, and then this is the nearby high school, this is a nearby K-8, and this is a nearby daycare, and then, you know, Portland neighborhoods kind of around the edges there. So the monitor comes back and they found air concentrations of cadmium and arsenic were 50 times and 150 times state health thresholds. And so it turns out that the facility had glass furnaces that had operated without filtration for 40 years, and that technically that hadn't been illegal. So this was our first collective, like, oh fudge moment, knowing that this is gonna be bad. So we, or rather the DEQ, actually immediately began reaching out to other key stakeholders. So the state and county health authorities in particular, together we worked on this painstakingly worded press release to help explain this odd situation to the public. And we began preparing for a couple community meetings at the schools nearest to the cadmium hotspots. But before that happened, somebody leaked our results to the press. So, you know, public response to our findings would have been bonkers regardless, but, you know, finding out through a leak just set off what I can only describe as a trust death spiral between affected parties, and especially between the communities and the regulators and the stained glass facilities, you know, and sprinkle in a little bit of like media frenzy and some lawyers, and we don't need to go into it. But long story short, there were about 6 months of chaos and extreme stress, and I still get, my heart still races talking about it. So I wanted to just take a second and take a deep breath, and maybe have a little humor interlude. I don't know how many folks are on social media, but every once in a while, a variation on a meme pops up that makes you feel so seen. Okay. Anyways, back to business. So just a few slides left before moving on. You know, we did have some opportunity at the community meetings and in media interviews to give some of the basic framework to help people understand what you could and couldn't say about the MOS data. The bare essentials, like, you know, hotspots on our maps are just the highest values in the data set. They're not inherently meaningful, and we can't infer health risks from MOS data alone, which was critical because we hadn't actually released our other maps of the other heavy metals yet. Anyways, it was really horrible seeing people so scared and hearing stories of odd illnesses in those neighborhoods. And, you know, people are still extremely angry, I guess. Just last year, the results of a public health assessment around Bullseye came back inconclusive. And so they feel they've had no closure. So I did what I could, you know, in answering every phone call and email, et cetera, to help, you know, facilitate an understanding of what we'd done and what it meant. So the bright side was really bright. You know, filtration was installed, air quality was improved in those neighborhoods. The national regulations for furnace emissions were strengthened, so this sort of thing wouldn't happen again. Increased funding to DEQ, more air monitors deployed, which is fantastic. The governor overhauled how we regulate toxics in Oregon, and some really keen community groups like the Eastside Portland Air Coalition cropped up and have been participating in every chance they get in the Cleaner Air Oregon program and hearings and such. So that was great. Finally, so 2 class action lawsuits were recently settled based on data from the study. One was Bullseye, and then the other case was less publicized. It involved an aerospace company in South Portland, where the MOS maps were showing really high nickel. This value is this outlier out here, and several other metals that were found in the alloys used at the facility. And it turns out that neighbors had long been convinced but unable to get much traction around. I mean, they've long thought that this facility had high air and water emissions of various pollutants. So there was this settlement, and the facility has improved its filtration multiple times. So some really good outcomes, I guess, depending on your role in all this. And then just finally, you know, another impact of this study was that a lot of people heard about it. So a lot of people have approached me, mostly people from community groups reaching out for help, wanting to use MOS to investigate pollution sources that they believe are harmful, but haven't been able to get much traction around. So we are working on a user guide to hopefully boil down some of the protocols to make it user-friendly for different audiences. So moving on to case study number 2 in Seattle. So this was my first experience with community science. This study was community initiated and community led in many respects, and it will guide a lot of the content we put in the MOS user guide. We have to figure out a better name, but that's what we're calling it for now. So in this study, we focused on industrial adjacent neighborhoods in the Duwamish Valley. You can see in this picture here some of the industry that lines the river. And so the Forest Service and the broader Federal Urban Waters Partnership have a long history of working with community groups and other stakeholders in this area on environmental health projects. And undoubtedly we benefited from that, some shared history and, some relationships were already in place. But we did still need to work pretty hard to earn the community's trust and vice versa, which I'll discuss in just a moment. So you can see maps of the area here. So the Duwamish Valley, it has a very complex social and economic history. It became Seattle's main industrial hub over 100 years ago. There's many legacy sources of heavy metals and other pollutants. For instance, 8 miles of the river, including all that intersects our study area, shown in yellow, as a Superfund site. The banks of the river include many soil contamination sites in various stages of cleanup. And there are also many active pollution sources, both industrial and transportation related. There are, highways and, there's a harbor and there's river traffic and there's a train and there's an airport all in that little area. So very complicated situation. Unlikely we're going to find, a really clear hot spot, like we did for the glass companies. So the neighborhoods that we focused on, Georgetown and South Park, are widely considered environmental justice communities. First, health disparities among residents are well studied. Examples include lower life expectancy, higher heart disease rate, asthma is also exceptional in terms of prevalence and severity compared to the rest of the city. Additionally, this area also has more people of color and people living below poverty level. So it's widely expected that air toxics are one of the drivers. And many model data sets would agree with that, including the kind of dispersion models that I was dissing earlier, that are based on knowing emission sources. But one key part of the problem, again, is the usual lack of actual air quality data. So the community inquired about conducting a moss study. So here's another map of our study area. You can see Georgetown and South Park kind of nestled amongst industrial land in this river valley that experiences periodic air stagnation events, as valleys do. There were 2 air quality monitors here and here, with only one of them measuring heavy metals, so not a lot of actual information on heavy metals. So by mapping metals and moss, so that's all the blue dots you see on the map, we were able to produce a first look at neighborhood scale patterns. So local youth-led moss sampling, which was a really cool aspect of the study. And we did, as a QA measure, have experts revisit some of their moss collection sites. And we were able to confirm very good statistical agreement between expert and community collected samples. I do want to emphasize that, the community, which was organized under the Duwamish River Community Coalition, was involved in all steps of the research, sometimes in a leadership role, and sometimes in more of a supporting role. It wasn't just that they, collected the data and then we took it from there. So here's just a few more action shots of the youth collecting and processing the samples. overall about 55 people participated in this study. And, let's just look under the hood for a minute. We got off to a rocky start, especially myself as lead scientist and the community leads. Communication was poor for various reasons. Part of it being COVID forcing us to really get to know each other over Zoom meetings where you can't really see all the visual cues you need to get a sense of people. And, it was difficult. We both kind of ended up being a little paranoid about the other. For instance, I wasn't willing to release the data until it had been QA/QC'd, which does take time. And I was still in a defensive stance after Portland, thinking a decimal placed in the wrong spot could be catastrophic. And what if somebody leaked that information, etc. So you can see where I was coming from. And then for them, that seemed like a big, powerful move, and that we weren't sharing. And from their perspective, it seems like in collaboration with scientists in the past, they often were left out or felt like the scientists had used that information for their own gain. So they were jaded for a wealth of reasons, which I understand. Anyway, so we formed a steering committee and an MOU that elevated the community's perspective and put them first. And this helped tremendously. While making this MOU was a giant time suck, it was critical for helping us foster the trust we needed to work together. So we all knew and created and agreed on the rules of engagement, including me sharing the data with all members as soon as QA was complete, whereupon the data became owned by the steering committee, and then we all would vote on all uses of the data, including major communications with the public about it. So this flow chart looks a little bit garbled, but the key ingredient to notice is that, the community had to support every action before we even went to vote on it. So I'll give an example of how this came in handy after we go through results. I can see I don't have a ton of time left. Okay, so number one, we found that heavy metals in the DV moss were significantly higher than in similar sites. So, in a nutshell, that helped the community decide whether to investigate metals further. As I said, there's a lot of potential air toxics of concern in this area. So we compared heavy metals in Duwamish moss, that's in these orange boxes, to similar data sets, including the data set from Portland, which you know about, and a data set from Seattle City Parks. So in all cases, the Duwamish moss concentrations were significantly higher. Also notice that for arsenic and chromium in particular, nearly all values in Duwamish moss exceeded the 95th percentile from Portland. So, this was a good sign to investigate heavy metals further. Number two, these maps provided the first ever visualization of patterns at the neighborhood scale. So, stating the obvious, that helped the community prioritize where to focus their continued investigation. And, maps provoke a pretty visceral reaction in people, a fact that you don't want to abuse, but, people are having a hard time getting attention for an air quality issue. Maps can sometimes be a catalyst for getting that attention. So we'll come back to that. So here are those maps of the metals in moss, and it's a lot to look at. The important part is, if you use principal components analysis, you can see that, a lot of the metals had similar distributions, being higher in the industrial core, etc. So finally, we found some correlations between soil and dust indicators. which suggested that coarse particles or PM10 might be important carriers of heavy metals. PM10 is not often measured, which was a surprise to me. It's usually only the PM 2.5. Okay, so quickly outcomes. First, there was a lot of social benefits to how we did this study, and that almost deserves its own presentation. Second, a lot of new funding was made available in response to these results. Third, and very importantly, with the MOSS results in hand, the community was able to advocate regulators for follow-up monitoring using air monitors. So that happened at 6 sites, which are the stars that you see here on the principal components map of metals in MOSS, which, is critical, because by actually measuring air concentrations, we start putting metals in the valley into terms that regulators and public health folks can relate to. And it also, helps start closing the loop, perhaps on, whether heavy metals are an important part of the health disparities being seen in the area. So next steps, and we do have the results of the air monitoring data back, and it's not mine to share at this stage, but the metals, including arsenic and chromium, do exceed cancer screening thresholds. So the regulators the Puget Sound Air Agency are now working on a synthesis report, which we should see in later in 2023. And there's some important outstanding questions like, how much of that chromium is in the highly carcinogenic hexavalent form, which we don't know how to differentiate in MOSS. So, there's still a lot of questions and such, but I do think that, by collaborating with community and working with this MOSS, we've been able to kind of advance their understanding of the environmental toxic burden in the area. So I know we're low on time. I'm thinking about how to say this more quickly. One of the biggest challenges, I guess, was just that we all come to the table in this project. There's a lot of us with like different motivations and goals and different skills. And I found it tricky as a person trying to do the science of it and trying to stay objective, working with people who have, a vested reason, understandably, to want to do things in a more like activist or sort of advocacy role. And ultimately, our MOU helped us hash that out and come together. But we did have a lot of issues where, people wanted to do different kinds of messaging. For instance, say, oh, well, all the metals in MOSS relates to this one point source that we've been, suspecting for a while, and wanting to, use that as the messaging. So anyways, I guess my point was just that, I had to be willing to let them go their own way, potentially, like make the case, like, why don't we keep it more objective and follow that way to resolving this, but also be okay if they chose to go off the community and use the messaging differently, to let them do that and be able to stand back and, not support it, but also not hinder what they wanted to do. Anyways, we ended up publishing some cool papers together, and we worked really well as a group over time. And then it seems like we're sort of out of time. Am I right?
I would go ahead and if you could maybe take like 4 or 5 more minutes.
Okay,
Just to
perfect.
leave time for questions.
Okay, perfect. So this one I just wanted to touch upon really quickly because we're really just getting started. So this study, Jason is also a part of. In this case, we are seeking to use moss to learn about air quality around 2 waste disposal sites in Oregon. This is also a community initiated project. So the picture on the left shows Eleanor, who's a very keen postbac who led the moss collection effort last summer. And here she is presenting the method of the study to the local community around one of the sites. She's been pretty fantastic. She provided me with this slide from that neighborhood event that I think captures our objectives pretty well. Besides learning what we can about air quality using moss and trying to get those finer scaled perspectives on pollutants, an interesting additional part of the study that I'm super excited about, which involves Jason, is the question is, can we look at different characteristics of the particles collected by moss to help us better identify the pollution source? That, for instance, would really come in handy in the Duwamish Valley study, where, the community wants to know, is it this metal recycler versus this or that? And so that is a really cool idea, because otherwise we're really basing a lot of our work on just spatial correlation alone. So basically, the study involves a medical waste incinerator, which you can see the location in the bottom left, in a transect of sites with increasing distance from the incinerator. So there's 21 sites that they visited there last summer. And then there's many more sites they visited here around a landfill further south in Oregon. And so this landfill accepts ash from the medical waste incinerator, as well as ash from various other medical waste incinerators and local trash, etc. And it's long been a big concern of neighbors in the area who say there's a cancer cluster and just some interesting health stuff going on. And so this facility, this landfill, is looking actually to expand and accept more waste from more places. And so everyone is, trying very vigorously to make a case against that. Final slide. So this field work was mainly done by Eleanor and the local community, which was pretty cool. I think they all had a really good time collecting that information. So unlike the Duwamish Valley, though, this is a very different situation. So at least around the landfill site, that's close to Oregon State University. So a lot of the community there are retired scientists and academics. So, they're kind of happy to let us do our thing with the data. And, of course, we'll discuss about what we see and this and that. But there isn't that sort of I guess you'd call it trauma, maybe from, people coming in and starting these projects, trying to help you and ultimately not, or, using it for their own gain. So, but the main active community group around the incinerator is an air quality rather air toxics activist group. And so that's a little different. And they've been trying to get that medical waste incinerator shut down for quite a long time. And actually, you know, the local community there that lives around is not participating at all. In fact, I'm not sure if they'd be pleased to know that this work is happening. So it's a very, very different situation. So stay tuned. You know, unlike the Seattle work, this has been pretty grassrootsy, you know, at the level of us like pinky swearing on a Zoom call that, you know, will share the data and be transparent and no one will go rogue. So anyway, stay tuned. This should be pretty interesting, and hopefully Jason and others will be able to maybe figure out some way to look at particles and say more specifically what their sources are. So that's it. That's the gist of it all.
Well, thank you so much. That was a super interesting seminar, and it's so interesting to see how, you know, others, what others' experiences are in community engaged research and sort of the adventure that entails. You know, I thought the look on your face and the photo of the press conference was very telling, you know, with your first experience. So in the chat, you know, Ellen, we'll start with Ellen. She has a kind of a comment and a question. Ellen, did you just want to jump in?
Yeah, real quick. Kudos, Sarah. Your work is just so inspiring. And to have your first case study turn into such a policy, regulatory, community mobilization success is like unheard of. So kudos on that. I guess let me just jump right to the air quality monitoring question. So do you know what kind of air quality monitor, and this is probably not what you do, so it's fine to say you don't know, but were the monitors that they put in the Puget Sound, the second case study, do you know what they were, what kind they were, and what they, do you know?
Yeah, so they're actually, they borrowed I had some monitors that I purchased that are near federal reference method, but not, you know, of course, as souped up as those permanent monitors, but they
Were they purple dust? Were they purple dust? No.
No, not the purple airs. They're very similar to, they basically are on these tripods and they suck in air at a certain rate through filter.
Okay.
So
Well, the reason I'm asking is because it looks like the PM10 was of interest, but from a health, I'm a nurse, so from the health perspective, you know, you don't breathe those in because your cilia, you know, filter the PM10s out. But my question is, were you measuring 2.5 at all? And is that the plan?
Yeah, so, you know, PM10 includes the 2.5. We did not in the Duwamish Valley area measure the 2.5 alone. We measured the whole fraction, including those coarser particles. So,
yeah. But from a health, my point is from a health perspective, you know, if you're trying to make that link into respiratory disease, right? Lung cancer and certainly not just cancer, sorry, respiratory disease, asthma, some of the other things these heavy metals are concerned, you know, would cause, I think you would have to measure PM 2.5. That's the smaller particle, because otherwise the other one does, I mean, not that it's not good, but from a perspective of the community and people who get sick from these things, I think, unless I'm wrong, it would be the smaller particles that would get into the lung.
Yeah, I mean, technically, what I've read in the medical literature is up to PM10 are the health relevant sizes, and beyond that, not so much. And then within PM10, those fines are more important because, as you're saying, they can get like much deeper in your lungs, etc., than the PM10. So, you know, there is precedent to look at those separately.
Yeah, I would encourage you to do that.
Yeah, they do have PM 2.5 in that area monitors.
And then I guess just to kind of chime in on what we're doing, you know, as part of the collaboration is actually looking at the particle size distributions and the particles in the mosses, so that may yield some insight.
I mean, the thing is, with PM 2.5, because they're much smaller particles, you don't really need quite that density necessarily to see the variation. So that's why we also include that larger fraction, because it is all health relevant within that range, and those PM10 and the larger than fines will tend to deposit closer to the sources and have a more heterogeneous pattern, I guess. Does that make sense?
Yeah, I mean, from the health perspective, I think it's if you can show that the moss or that, okay, so like you said this so well, that moss will capture the heavy metals, right? We know that. We just don't know if it's going to translate to human health necessarily. And then you said, well, it's in the PM10, that PM10, if I understood you correctly, carries, right? Like they carry the metals, which is really important. But then the question is, you would need to, I think, measure PM 2.5 in the environment. And then I think Jason's work's important because if the metals are on the 2.5, then that could be carried into the body. You know what I mean? Because 10 won't be. I mean, it's not going to cause the kind of chronic disease that we know is caused by these metals, like specifically arsenic and cadmium, of course, because of cancer and respiratory illness.
Mm-hmm. Yeah.
Well, I guess, I have a question that kind of is related to this. So I'm not so sure I agree that only the finest particles are health relevant. And that's because you have deposition of pollutants into soil. And then ingestion, particularly in children, becomes an important route of exposure. So I guess I was wondering, like, for example, in the Portland study, has there been any follow-up sampling, of soil in those areas? So, even if they've reduced the air emissions, there could be legacy pollution in the soils in the area, particularly where you have schools and things like that, and then tracking dust into the homes. Do you know if they've looked into that or?
They did, and there had been a big announcement that went out to people in the area. Say, a lot of people grow food gardens, saying don't eat your vegetables, and so they did a lot of free soil testing, and of course, right up on the facility, the soil was so bad that they actually had to come in and remove it and dispose of it properly. So there is some legacy in the soil, and they did look pretty extensively to figure out, where cleanup would be required. Does that answer your question?
Yeah, I guess that really gets at what I was asking. I think I kind of was seeing it as the moss kind of indicating where you have deposition, and then further investigation would be required, to look at routes of exposure after that.
Yeah.
Do we have other questions from the audience that aren't in the chat? If not, I guess another question that I have is, how does the Forest Service leadership kind of look at using citizen science data? So for example, in some of the watershed work that we do here, the Division of Water doesn't really look at citizen science collected data as actionable. And I was, how is that why they have to bring in the, instrumental sampling techniques to get actionable data and things like that?
I mean, the instruments, the moss just doesn't translate directly into any health thresholds, or it
Yeah.
it reflects like air concentrations over time, but it's just not exact enough that I think it could really operate without having those instrument values to kind of connect it back
Mhm.
to health. That said, the Forest Service at least is really into this community science and funding it well and supporting it well. And it seems like the EPA is also quite a bit. Granted, you still need that follow-up step, they would argue, of actually monitoring what's in the air, but they've been hugely supportive, which I think is neat.
Yeah, I was going to mention, I've been hearing rumblings of the EPA having a big push for citizen science. Jason, Marion, did you have a question?
Yeah, I'm happy to ask one. I love that presentation, by the way. There's so many applications, so many communities would love to use it. I would be curious on the look back period, like how far back does the moss actually, go back? Is it related to, we've got older mosses on trees, younger mosses on trees, the age of the trees and so forth. And I'm just curious about the look back period.
Yeah, I actually can answer that as of recently. So after the whole bullseye thing happened, and people leaked the information, and then we were trying to, through all that noise, help explain to people what the moss said or didn't say. One of the questions is, well, what time frame is represented? And it didn't really know an answer. So I was like, probably a few months, or we don't know. And so we got funding to do a calibration project where we set up these sites around Portland, and we sampled the moss every month, and we had a PM monitor, and we had both deposition monitors. We looked at PM10 and PM2.5, and tracked that for 14 months. And so I can tell you that it reflects about a year. And just the way, I don't know quite how long lived this moss can be, but the way we collect it and trim it up, we trim away kind of some of the older dead parts and keep, the greener tips. And so, that in a way sort of standardizes what time frame you're looking at. So for instance, there's some folks from Keith Western that have been asking me, should we go out to that area in East Palestine and try to look at dioxins and moss or lichen out there, and would that be useful? And I guess I'm not sure how well it does with acute events. Well, that one seemed like it was pretty acute, but did I answer your question?
Yeah, you definitely gave me the things I was thinking about, because I know some people may want to look back 30 years or 20 years, but I know it'd get noisier. So, but I think your question was excellent in terms of the methodology and following a standard method. And I look forward to seeing your moss guide for the future.
Thanks.
Well, we're a little bit over time, so I just want to make sure that we thank Sarah for a wonderful presentation. Very interesting work, and it seems like has already had a huge impact. So just really impressed. And then, so yeah, thank you so much. And then also I wanted to mention, Sarah, I do have an idea of how we could differentiate between chromium-3 and chromium-6 in the moss.
Really?
So maybe that's something we could talk about when we meet.
That would be incredible. That would be super incredible.
So, all right. Well, thanks so much, everybody. And look forward to seeing you again soon.
Thank you.
Thank you.
Bye.